Have you ever seen individual atoms, weighed individual cells, or watched a natural nanomachine at work? All of these things are possible using a very special microscope known as the atomic force microscope. When we hear the word microscope, most of us will picture something like this. As children, we might have used them to observe the life in a drop of water and marveled at all the things you find swimming around in puddles and ponds. You might also have seen images such as these from scanning electron microscope and discovered that the dust mite with whom we share our homes isn't the cutest of creatures. Very few of us will ever have seen individual atoms or molecules under a microscope. Researchers can produce images like these using scanning probe microscopes, which look very different dependent on their application. The microscopes come in two fundamentally different types. The scanning tunneling microscope is suitable for conductive materials. With the atomic force microscope, on the other hand, it is also possible to analyze biological samples in their natural environment. And that is precisely what the atomic force microscope, or AFM for short, has done so successfully. Researchers from Zurich and Basel played a key role in driving forward the development of both types of microscopes. Professor Christoph Gerber is one of them. Together with other researchers, Gerber has received numerous prizes for the development of the AFM.
This here is a replica of the first atomic force microscope. It is in a permanent exhibition called The Making of the Modern World in the Science Museum in London. The motivation was to develop a new microscope that primarily images non-conductive surfaces at the nanoscale. The idea was to measure and control atomic forces acting between the tip and surface at very close distances. The heart of the microscope is a microscopic cantilever with an integrated sharp tip. In very close vicinity, the surface to the forces bend the cantilever. The deflection is measured by a laser, and from this the force can be determined. The invention of scanning probe microscopy in the early 80s led to a shift of paradigm in the understanding and perception of matter at the most fundamental level. The first cantilever was made from a micrometer thin gold foil, and the first tip was a fragment of a diamond crystal from an old turntable glued to the cantilever. Not necessarily for a proof of principle you have to build a Ferrari. The whole overall cost for the microscope did not exceed $100.
The invention of scanning probe microscopes made it possible to investigate the nanoworld in the first place, and the development of new instruments and applications continues, including in the world of physics, for example.
So we measure with the AFM on single molecules. We can image each atom, get intramolecular resolution. In addition, we can also move the molecule along the surface and measure friction, which is a phenomenon to investigate energy dissipation on atomic scale.
In our case, we use AFM tips to place individual electronic spins in close proximity to objects of interest to use them for quantum sensing. One particular application is the study of atomically thin magnetic materials, which will form the building blocks of future high-density memory devices.
The AFM is also particularly well suited to addressing wide-ranging questions in biology and medicine.
We try to understand how molecules and cells of life interact with each other. Here is a particular application of the atomic force microscope where we measure the weight of the cell, which is important if you want to understand how cells grow into larger cellular systems like tissues, organs, or animals.
So we use the high-speed AFM to film biological nanomachines at work, and in particular we're fascinated by the molecular machinery that regulates transport in and out of the cell nucleus.
Our team applies AFM technology in clinical diagnostics. We can detect bacteria and antibiotic resistances in sepsis. The sensitivity of our device is in the range of 10 bacterial cells per milliliter blood.
Given that the AFMs also come in compact form and as portable models, they can now even be used by school pupils to analyze micro and nanostructures. The technology has come a long way from the first AFM prototype to today's high-tech devices, with several key milestones taking place in Switzerland. And thanks to the work of countless researchers around the world, this journey is far from over.